Okay, let me try once again. So, do we have anybody from Pittsburgh? Ziv, are you there? Anybody from Cell Signaling? You there? No. From Washington, are you joined by any?
Hi,
Hello.
this is Ziv from CMU, and my group is here, but they're trying to connect. They'll be on in a few seconds.
Okay, great. So are you having troubles connecting?
No, we just didn't realize we needed to download the application, I guess.
Oh, okay, great.
No, it's fine. Are you able to?
I'm working on it.
Oh, I hear you. Yeah. Awesome. I think we should turn only one speaker on, otherwise this will get confusing.
Okay.
I'll turn it off. Can you mute it? Yeah.
Okay, well let's start. Hopefully people can still join in. So the purpose of this conference again is to basically ascertain how you guys are doing in getting data, processing data, and using data, and whether you need any help with that, and to a bit of an idea of what kind of process data may be available to you or may be made available to you from our three centers or three groups that have been working on this data for a while now. So can I ask a couple of questions just before I start? I would like to ask somebody from University of Washington if you were able, whether you were able, maybe Chad, whether you were able to actually access data and do anything with the data that is available through Linux Cloud. No?
Yeah, I don't know if... Oh, Chad is there. Okay.
I'm just going to say I didn't actually hear the question.
Oh, I'm sorry. So I'll try to speak louder. So I like to see, just get a little bit of a feeling how far have you progressed in actually being able to use or download the data, because I have a little presentation on how to do it. If you guys already have the data and have processed it, maybe I will skip some parts of this presentation.
Yes. Is that Hong?
Yes. It's Hong. I'm here. I can speak. I've been able to actually get. Hello. Can anyone hear me?
I think somebody did that with.
Okay.
Okay.
Can anyone hear me?
Yes.
I can hear you loud and clear.
Okay, good. So I've been able to download the large GCTX file and parse it to get the different data slices out and to relate it to the big Excel table. However, what I think to all of us is that the identifiers for the gene names in the GCTX file don't seem to match anything that is recognizable. Is there a table for that to map that to something like Ensemble IDs or whatever you are using internally?
Okay, I will address that then in a. So, let me just ask a follow-up question. So you were able to parse the GCPX file, but have you navigated the APIs that they have provided for accessing and querying metadata on these signatures?
I just used they did give us, there was a raw file with like 32 fields with, it seemed like it had all the data for all the perturbations. And I just used that in order to relate that back to the GCTX file. So I assume that's the majority of the data. There's some questions I still have about that, but I can't seem to find anything about to relate the links 1000 set, which you have in one table, and you give 3 identifiers for, but none of them match the identifiers for that are used in a GCTX file.
Just to add to that, I have been able to use the API, and I've kind of explored around what I could in there, and I've been able to find at least like I think it's the file name that matches for the perturbation or whatever it is, and so I have done some exploration on that side of things.
So, okay. So to give you a short answer to the first questions about the genes, so that's great, and I'm glad to hear that actually you guys progressed more than I thought you would. So the identifiers in GCPX file, I assume you downloaded the level 4 data. Am I correct? Or
level 3 data.
Okay, level 3. I believe the identifiers are AFI probe set, and there are actually there is a web service, one of the web services that is available through the APIs, gives you information on the complete annotations of these probes, probe identifiers, so they match them with
I was able to find 1,000
and trans IDs.
Okay, so there is an API where I can send all 26,000 genes and get an identifier back.
That's correct.
Yeah, that particular page is called gene.
What is the gene naming scheme? Hello?
Yes.
What is the gene naming scheme again? You mentioned IDU something.
It's an Affymetrix probe set IDs.
Okay.
Oh, I see. Okay.
Which array?
Affymetrix. Oh, which array?
Yeah, I don't recall exactly, but the reason is the way historically how this assay works, and how they derive their model for estimating gene expression levels. That's why it's based actually on a large Affymetrix data set of public domain expression data, and that's why they are still annotated in this way. But this is the easiest of the problems to solve. I will point you to the API, to web service, where you download all the information about the genes, and I will be also happy to share a simple table that maps if you don't have it available.
Yeah.
That would allow you to do that. So I guess both groups then were able to download data and parse the data, and Ziv was also able to use APIs to download the metadata.
Yeah.
I guess that will shorten my presentation. So one last question: Did you did any of you actually progress to any degree in actually constructing in constructing signature or aggregating different replicated profiles for a specific perturbation?
We didn't.
Okay. So that was Ziv. You said you didn't, right?
Right, yeah, we yeah. Oh.
So maybe mute everybody except for me.
I have muted my speaker so it doesn't like reverberate everywhere.
I do have one
You can't hear
question
me?
about
Okay.
that. The perturbations, I noticed looking at the table, there are a set of different controls for them, and they're a set of there's a fairly large set of data associated with each perturbation ID. Is there a set organization to how that was done? Like, are there a certain set of controls for each, sort of like an experiment for each perturbation that we should
Right.
look for?
So let me now start with this little presentation that I had, and it's going to address a bit some of these issues about how exactly to normalize data and how to use whether to use controls or whether to do a global normalization per plate, and what Broad Institute recommends, or what's their philosophy. And then later on, there will be a presentation given by Mayan group about how to use actually controls on each plate to construct or to aggregate these data. So the short answer to this, yes, there are controls that you can use. There's also a way to do that without actually comparing perturbations to the controls. So it's kind of one of the unanswered questions about this data is what's the best way to do that, and maybe depending on the situation and the specific analysis that you're performing, there may be different answers to this question. So so my intention was to tell you about getting data, metadata, and signatures, and but since you kind of already made progress in parts of this, I will skip a bit through this presentation. So let me go forward. So this is parsing the big matrix, and I assume that you would actually start with the level 4 data, but that's okay. Level 3 is fine as well. And you parse that. For some reason, do you guys see?
So, Mario, just the screen sharing is paused, it says, so we can't see any slides.
Sorry, I apologize.
Okay, there we are. I can see it now.
Okay, thank you. So let me give you a bit of a background, just quickly, in terms of how this data is generated, and that may answer some of the questions. So I want to acknowledge that I stole these slides. I didn't steal, I borrowed them, and he agreed to lend them to me from Arvind Subramanian from Broad Institute, who is, I guess, Call
ended.
What?
I'm still here.
Okay. All right. I heard call ended. I assumed it kicked me out, but it must be
something. No, you're still there.
Okay, great. So the story starts here. So basically, by looking at So I disabled your mics for a second, just to calm down this feedback. I will enable them now again, but please be cautious. All right. So,
by the way, hello?
yes?
Hello?
This is Peter Hornbeck here at South Signaling
Yes, hi, Peter.
with Steve and Ben.
Hi.
Hi.
Thanks for joining in. I'm kind of starting with a story about this assay and how the data looks like and what it means, just brief intro. So it was a good time to join in, all right?
Yeah.
Can you see it well, Peter? Can you see that?
Yes, we can.
Yeah.
We'll have a lot of questions because we're just new. Yes.
Okay.
I think we need more sound now, though.
So,
Right. I think for some reason they don't pick up. He's not talking yet.
So by looking at this big picture, you see that a lot of genes are correlated, they form clusters. So the idea of this assay is that you don't really need to measure expression levels of all genes. You can measure expression level of genes that describe major patterns of behavior and then estimate other genes based on measuring these specific landmark genes. So these are going to be the landmark genes that are being actually directly measured. So the approach that Links or L1000 assay is taking is that they measure, and basically what Broad Institute did, they did benchmarking and established that 1,000 genes would be sufficient in order to recapitulate behavior of all genes in the genome. And so the idea is we measure expression level of these 1,000 genes, then have a computational inference model that uses expression of these landmark genes to predict expression levels of all of the rest 21,000.
So can you say something about the computational inference model?
Can you repeat that? I didn't hear it.
Can you tell us how the computational inference model works?
Oh yeah, well, it's a simple linear model, really. You kind of have a big matrix of coefficients, and it's derived in somewhat robust form, so that it's relatively sparse, I guess. And you plug in for each gene in the genome, you have a regression model that has as the variables the measurements of the landmark genes, and it's based on the analysis of these.
Sorry. Hello?
Yes?
Yes, I have a question. Someone is typing very close to a microphone.
Okay, so please don't type close to the microphone. Thank you. So the big data set that we use for deriving the inference model is consists of 100,000 asymmetric microarrays, and that's where the probe set annotation comes from in actually calling or naming these rows of data in this GCTX matrix. So the model was derived based on this big data set of microarray measurements, and it says here about 100,000 profiles. So landmark genes are these 1,000 genes, and again, one of the annotations that you can download using these web services is the identity of these landmark genes. So the bottom line is here. By measuring only 1,000 expression of 1,000 genes versus the whole genome profiling, the cost goes down about 100-fold, and the throughput also increases significantly. So using this assay, Broad Institute was able to generate about 1.3 million genome-wide expression profiles of cellular perturbations over a 3-year project, during the 3-year project. So in case you didn't know, the major perturbations that are being performed are gene overexpressions of about 3,000 genes, knockdown of about 3,000 genes using 3 different shRNAs for each gene, and then treatment with 4,000 small molecules. This is all data. The data matrix of these different perturbations is bigger now, but these are the ballpark figures.
Can I ask a question?
Yes, absolutely.
So how are these genes selected for the overexpression and for the knockdown? And is there any plan to complete it?
That's a good question. I don't think so, actually. I don't think that in the second stage of the project, the plan is to complete and to have all the knockdowns and of all genes in the genome. This number did go up. So there's more than 4,000 perturbed, but the plan is not to do the exhaustive perturbation.
So I have another question. We looked at some databases, and some of them show 16,000 or even more genetic perturbation, and you only list 12,000 here. Where are the other 4 coming from, or 4,000?
Right. So as I said, these are old numbers. So they have the matrix is not complete. So for example, there are close to 50 cell lines that were profiled under at least some perturbations, but not all perturbations are profiled for all cell lines. So I think if you look at how many genetic perturbations have been performed in at least one cell line, you get a much bigger number than, say, how many perturbations have been performed in a close to exhaustive way across, say, all 15 or 20 primary cell lines that were originally used. So I think your numbers are correct. There are more perturbations. There are more. So the number of shRNA knockdowns, this is also a ballpark figure or lower kind of lower limit on how many shRNAs per gene are used. So many genes have more than 3 shRNAs that have been used for targeting.
Mario, let me add something. This is Avi. One of the things that we learned from that data, from the knockdown data, is that it's not that good, and the Broad acknowledged that the shRNAs that they use, they're not very good in eliciting the expected responses. So they're moving to CRISPR-Cas. So they're going to redo knockdowns rather than expand in the next phase.
Right. Can you hear me?
Yes.
Can you hear me? Okay.
Yes.
This is Peter Hornbeck. I also wonder, I suppose this is published so I can read about it, but I do have very basic concerns about the whole method of being able to use only 1,000 genes as a reporter to try to predict the expression of every gene that you're going to be interested in, or what? Or are we going to only look at these 1,000?
Well, I mean, there are situations in which looking only at 1,000 will give you about the same level of information as looking at all of them. But also there are situations in which you would want to look at predicted expression levels as well. And it is a good concern, and the limits and of this strategy are, I think, still to be established, and there is no paper at this point yet in terms of describing or benchmarking this strategy. To the best of my knowledge, Avi, correct me if I'm wrong. But the main paper that was supposed to be published a while ago still did not come out.
Thank you.
Okay.
Mario, this is Lucifer. I think the original idea of reducing the whole genome to just like a number of representative genes was done and published a long time ago.
Oh, it was?
Yeah, but they used the gene set enrichment in that paper. And my understanding is that for the links, for the pilot phase, they used 68 to reduce the number of genes, 1,000. So they still show that these 1,000 genes can represent 80% of the whole genome. So the idea is to reduce the cost and to reduce the time.
Right. All right, thanks. So,
Avi, Avi, can I ask, the overexpression is okay? Just the knockdown is a problem?
No, the overexpression is actually worse than the knockdown.
So this is Adrian Raftery here. I have a question about the nature of the result that the landmarks give you information about all the expression, or very high proportion of the information about expression of other genes. I'm wondering, does that extend to the network properties? So essentially, we're trying to infer network properties and pathways and so on. So if we're missing 95% of the actual information, has there been any assessment of whether just imputing the other genes will preserve network properties? Because if you've got, say, A is linked to B, is linked to C, is linked to D, is linked to E, and you're kind of interested in the links between the relationship between A and E, has there been any assessment of whether, and B, C, and D are all missing, whether the information about those more complicated network links gets preserved?
Done this kind of assessments. I'm not aware of publication that would talk about this. Avi, have you done anything of that sort?
I would caution about the single gene correlation.
Yeah.
But I would definitely think that the overall structure is preserved. So at the pathway level, and so assume that you have about 5 to 10% error compared to microarrays.
Okay.
So, and you don't really know, for the infer gene in particular, you don't really know if those are accurate. So you have this uncertainty that is increased. But overall, you do have a pattern that is, mostly consistent. So the method may need to be adjusted, not to rely too much on a single gene sort of like network inference, but sets of genes or clusters, and somehow address this level of uncertainty in the model, that there's also batch effects that they have to deal with. There is other different sources of noise that potentially are introduced here that can be potentially subtracted, and just because we haven't really worked with this data long enough, we can't really tell how we can extract the most amount of information from it. But I would be very careful with the Bayesian gene-by-gene inference type of methods that are used in the past. I don't think they're that good for microarrays or RNA-seq, and I think here it's going to be even worse.
Okay.
Avi, in the new awards, are they also focused on these 1,000 genes, or the proteomics, metabolomics are going to do genome-wide?
1,000 genes from the Broad, but there's going to be
and
some
and the other centers, like the Mount Sinai, Stanford, whatever?
There's some RNA-seq, and there is proteomics, but
That's
in general,
genome-wide?
Yeah, I would be worried about like genome-wide proteomics. Like there's some of it, but it's not in that scale. So you'll have many more columns or many.
No, but the question is, like, the new centers, are they going along with this 1,000 gene representation, or each center is doing whatever it wants?
Yeah, they're doing other things. They're doing a lot of imaging, and they're doing proteomics. They're doing a little bit of transcriptomics. They're doing multi-layered type of profiling. So less conditions. But
yeah, but again, Avi, but is there this focus on this 1,000 gene representative, or that's dropped from the next phase?
It's continuing with the Broad. So the Broad is one of the 7 or 6 sites that are going to produce data.
Okay.
So, Avi, this is Peter. Hello?
Yeah. Hi, I'm here.
Good. So the data that we have seems to me to be destined to be integrated with the proteomic data and not the L1000 data. Obviously, we're going to bring them all together at some point, hopefully. But is that correct? That we're going to be integrating our data into the proteomics side?
Yes. In principle, yes, but it can all handled sort of like the same. I think it would be good maybe to have another meeting where you describe your data to the other data science research groups, and because they're interested in analyzing that type of data that you have. So maybe they can have access to that data and then also work as part of their project on your data, which is probably very attractive to them once they understand what it is.
Right.
So, I mean, there is, if you guys are interested in integration, there is another data set that has been generated during the pilot stage, which is P100 phosphoproteomic data set, which has some of the overlaps in terms of perturbations, chemical perturbations, with L1000 data, and it could be used as complementary in any kind of network inference model, and that could be another topic of conversation, I guess, at some point later. I mean, it is listed on Linux Cloud, so maybe some of you have already seen it, but I'm not sure if you developed it.
I still haven't been able to get in. This is Peter Hornbeck.
Oh, okay. Well, so you applied for access, but still were not granted?
Well, yes, because we're from a company.
Oh, okay. Well, I think that's where my powers stop. Because
well, they don't know. They didn't know that we were associated with the Linux project, however.
Okay.
Yeah, I'm pretty sure they can make an exception.
All right, so let me just go a little further. I'm not quite sure how much you guys know about different levels of data that are actually available, and these big matrices that are available. As I heard, you watch people download the level 3 data, which is Q normalized data. So let me just give you a little bit on how exactly these level 3 and level 4 data are created. So everything starts with plates, 384 well plates, and data normalization and inference is done by the plate, really. So once these basic measurements are extracted, they are performed quantile normalization of the data per plate, and then the inference is performed to infer 21,000 measurements based on 1,000 measurements. So per plate, now we have, for each plate, we have these measurements, Q normalized measurements of all genes in the genome, kind of. So the next step that they perform, or the next data processing step, is to calculate these ZMAD scores. And you can consider these to be substitute for the treatment versus control type of differential expression level. So basically what is done, if each row represents expression level of a gene for 384 columns on a given plate, the data is basically Z for each gene is Z transformed by subtracting the median level of expression for this gene on the given plate and divided by mad estimate of the standard deviation for the expression levels of this specific gene on a specific plane. So the signatures that Broad Institute or their method for constructing signatures, final signatures by aggregating replicated measurements into a single signatures are based on these ZMAD scores.
So I have a question.
Yes.
So this only works if the plate experiments are completely independent. For example, if you do the same cell on one plate with different perturbation, that doesn't work. That doesn't make sense at all. This only works if you completely randomly place things on the plate, which I doubt they do.
They don't. So yeah, plate each the cell line is same for all wells in a plate.
So what if you have a gene that is highly expressed in one cell but not in the others? Then it will be the same z-scores for all of the different cells because it's always normalized to whatever expression it has. Why does it make sense?
Well, it makes sense because you're interested in different perturbations. So the genes that doesn't change across different perturbations, you may say it's not interesting, right? Despite the fact that it's highly expressed in this cell line.
I don't know, some genes may be highly expressed in one cell but not in the other, and a drug works on one cell and not on the other, and you will never find out that this gene is relevant.
Right. I mean, not from z-scores. So from z-scores, you will not be able to assess which gene is highly expressed in which cell line. But again, you can, for this particular purpose, you can obviously go back and look at the just normalized, Q normalized measurements. But I guess the objective here is to establish the signatures of a perturbation, right, associated with a specific perturbation, in which case you want genes that change, that are different after a specific perturbation. And that, that can work across different cell lines as well, because some genes may be perturbed in a similar way across different cell lines for a specific perturbation, and some may not. So it still kind of gives you information about which gene is affected by specific perturbation. So, I don't think it's the whole story, and I agree with you that one important piece of information is whether the gene is expressed and to which extent in different cell lines. But again, this is designed specifically to assess the changes in gene expression associated with the perturbation. Okay, well, anyway, this is the one thing about this data is that there is really there are some baseline approaches to processing in the analysis and constructing of these signatures, but many of these questions are still open. This is just kind of state of the art. So once you have these Z-scores, and each plate basically is replicated at least 3 times, theoretically at least. So you have, and these are called biological replicates because they start from the fresh saline cultures every time, and then they do treatment.
I'm using those.
But the replicates are done on the plate level, so the whole plate is replicated 3 times. And then the question is how to integrate these replicates into a final signature or summary of perturbation changes or changes in gene expression due to a perturbation. And the way that Broad Institute advocates that this should be combined is using this weighted average of Z-score, where weights are derived from pairwise correlations of the whole profiles between different replicates. So this is the moderated Z-score. So the idea is that if, for whatever reason, one of the replicates didn't work well and doesn't correlate well with other 2 replicates, for example, we don't want to put too much weight on these measurements because it could mess up our overall estimate. This would be the final, basically, product in terms of the final signatures of perturbation effect. However, these are actually not provided in the data that Broad makes available on their links cloud at this point. So the level 4 data are the Z mods, or these Z scores for individual replicates. There are 1.3 million columns or 1.3 million vectors of these, but the final signatures are left up to you to construct. So this is what Broad suggests as a strategy, but there are different ways to do that, and I can tell you about how we constructed signatures for that are in this iLinks package, which I know at least Kai took a look at. And the Miami group has their way to average these or aggregate the replicates. Avi's group has developed a brand new approach for constructing signatures based on these individual replicates, and we'll have a short presentation of how they do it, maybe if we have time. So the other thing that I would like to point out, I emailed you the link to the yesterday's meeting that we had. It's a YouTube video, basically recording of the whole session, and Chandan from Avi's group has presented this yesterday. So in case we don't get to it today, you can go back and view his presentation from yesterday. All right. so let me try to just go through this. This is unneeded because you guys already processed the data. So let me just finish this with the way how we constructed signatures, and then I will turn it over to Miami Group or the to tell you a bit how they are doing, or maybe we can have an open discussion, whatever seems to be desired. So the way we constructed signatures, instead of looking at these weighted z-scores, we do use pairwise correlation to tell us which replicates to use. And if we actually have a cutoff of 0.2 for, so if a replicate correlates with a correlation of at least 0.2 with all other replicates that we're going to use for just landmark genes, then we use them in constructing the final signatures, and the final signatures are just the averages of the z-scores. And we also construct p-values for these signatures by performing basically simple t-tests against z-scores being equal to 0. So our signatures consist of two scores. One is a vector of z-scores, these averages z-scores, and then the vector of p-values associated with it. And just going back quickly to the controls on each plate. So on each plate has several different control treatments, and you can use them to construct differential gene expression profiles for perturbations, but these z-scores that are advocated by Broad Institute as being a good way to summarize data, don't use this information, right? So they just kind of normalize against the population control, which is all measurements for a gene across all perturbations. All right, that's all that I have that seems to be meaningful at this point. Should we entertain some more discussion, or should we go to another presentation from maybe Miami Group or from
This is Peter Hornbeck. I need to go to another meeting, so I just had a quick question. It's off topic, off L1000, and the question is: will other disease expression data sets be available to us other than the L1000?
The data sets that are associated with the LINCS project or generated by LINCS project, or data sets that we gather from public domain repositories and projects?
Well, public domain I know is available, so I guess it's from the LINCS project. Will there be, for instance, the kind of data we have is really proteins talking to proteins, generally speaking, and tying that to the genetic expression regulation is a whole different ballgame. So we'd be interested, I think, in expression, disease expression of proteins in various disease states, and I know there's public data available on that. So is there going to be any specific LINCS data that will be associated to that in the near future?
Well, the data that comes to mind is maybe some of the data sets that have been generated by HMS Center during the pilot stage. And
yeah, if you're mainly looking for kinases or kinases and stuff, HMS has already generated some of these data sets. Like KinomeScan, Kinative, those are different essays that are right now available from HMS.
I saw that, but I hadn't been able or hadn't gone in there yet to explore what they have.
Yeah.
But I saw on the HMS.
Yeah, there is also some secretome, some data that is produced from AL, which looks for the antibodies and stuff.
Right.
Expression of antibodies in the cancer cell lines and stuff. So there are like and few other assays which are not normalized at this point, but still they have it. Like there's one which does like deep proteomics, which sees all the proteins that are expressed in like TCT cells and stuff. So there are some assays going on, but it's I think it's not available right away. I mean, it's not normalized and stuff, but I think it will be available.
Right. I know I saw Steve Carr was somewhat skeptical. I guess they did publish a paper on looking at the possibility of damage to the proteome during sample treatment. Anyway, and I think that data might be available soon. Or is Steve Carr? Can we communicate with Steve? Is he involved? He's involved in this LINCS program, yes?
I don't know that information.
Thank you.
So, right, that having said that, the HMS Center has generated a fairly diverse repertoire of these data sets, and it's kind of difficult to have them summarized in a comprehensive way. So if you would like to inform yourself about what is available there, I think the best way is to just go to their website and see they have a fairly nice interactive system that allows you to browse their data sets and you can see what is available and what could be made useful for your purposes.
I think in our website we have both from data from HMS as well as the LINCS Cloud in live.
So, Amar, would you want to spend maybe just a few minutes to then say something about live, what kind of additional information maybe can be accessed from live? And unfortunately, I think we're running out of time. So, Chanan, maybe you can present either at the next meeting, probably the next meeting, or a next conference call that we have scheduled, if that is okay. And again, if you want to see Chanan's presentation from yesterday, you can maybe take a look at the video. So Amar, I'm going to switch to you, and you have a few more minutes, and just if you could briefly go through what you have in the list.
Sure.
Is everything okay?
Yeah, it's hello. Can you see my slides?
Is that not working? I passed the presentation roll to you a couple of times and nothing seems to be happening.
Yeah. It doesn't go anywhere after.
Okay, so in the meantime, while we are waiting for this maybe to work, if you guys think that you are slowed down by any step of this data processing, and you would maybe want some of these annotations or some of these data sets in a pre-digested form, maybe you could contact me or somebody else, one of the other 3 centers, and we can maybe share some of these in a pre-digested form, table form that kind of can. That's one point that I wanted to make. Amar, can you comment on the state of your presentation?
Yeah, I'm not able to connect for some reason.
Okay.
I didn't know again.
All right, so our time is up anyway. It's 4 o'clock. I would like to wrap it up in time. Let's plan on meeting in 2 weeks. Maybe we can continue discussion with Amar showing you what is available from their system, in their live system, which basically gathers and annotates most of these data. And Shonan can tell you about his method for generating or summarizing replicated data such as signatures, and some of his benchmarks actually that he performed and showed that this may be the good way to go. So if nobody has any additional questions at this point, I would like to conclude this meeting. All right, any questions, final thoughts? Please let me know if you think this is not a good way to communicate. I guess I thought that this would be a fairly straightforward interface to use, but if it's not going to work, we can maybe try something else. I am looking forward to talking to you again next for the next meeting, and if you have any questions, please.
Mario, I think it was a good session. I think we should have a set of conversations like this. So maybe we should schedule the next meeting sometime for next week. So maybe Amar can have the full hour to present, and then Shonan can present the following week or something like that.
Okay.
Yeah,
so
I think that.
I am open to basically having it at the same time next Tuesday, if that seems acceptable.
Any chances that Arvind join us?
So Arvind will not be available probably to join us next week. He offered to join us sometime possibly in 2 weeks from now, but not on Tuesday, or then on the Tuesday at the beginning of November. So I will try to schedule something that will work for Arvind as well, to give us a presentation. But next week, I don't think that's going to work. So next week, we can still have Amar, and then the following week, plus the discussion. I think having this open discussion is extremely important. So maybe we'll have Amar present and then leave some time for discussion. Okay, so I will send the email to everybody, but let's count on basically meeting at the same time next Tuesday.
All right.
you.
Bye-bye.
Thank you.
Thank you. Bye-bye.
Bye.
Thank you.